I'm most excited to learn and see kind of how we can help scientists in the science community better relay the research information kind of to the public.
At the beginning of this innovative SciXchange experiment, I honestly didn't know what to expect. Joining 2 other student journalists, we quickly adopted the scientific method and let the learning commence.
I realized I'm not going into the field of putting plant cells into yeast, so I'm probably most anticipating learning more about the scientific process in general, because that's something that is as alien to me as microcentrifuges are.
The first step of the scientific method involves identifying the problem and coming up with a question that can be tested through experimentation.
First of all, we are not trained to talk to the public. We are trained to do science. So we have a communication problem, right? So on one hand, we need to learn how to communicate better.
With SciXchange, it's writing and observing. With Dr. Mendoza, we wanted to do was actually get journalism students and get their hands dirty. So have them do an experiment in the lab and then communicate about it in whatever way they're comfortable in, whether that's stories or blogs or videos or some combination therein.
The major challenges we are facing how to communicate with the outer world, basically to the non-science. When we communicate to the journalists, it's fine, but when we communicate to the non-science, then we are facing challenge how we communicate in a better way, in a simple and basic way, so that the non-science people can understand what we are doing and what's our major goal and implicate in the human welfare as a whole. So to project ourselves to be simple and in a better communicative way, that's you are coming in.
I think there should be a natural connection between the scientist and the journalist, and this is what I'm trying to do here. Not solve a major issue, but just the little issues, which is the technical aspect, right?
Before we could come up with our hypothesis, we had to do some background research to familiarize ourselves with transgenic organisms and the scientific process itself.
We are basically working on how the metals, movement of metals, or basically the micronutrients, how move from the soil to the seeds. And seeds and the plants and seeds, are basically the important dietary nutrients for all the human beings and also for the animals.
This is a great chance for journalism students to see all the time that it takes and to get that kind of inside knowledge. And I'm hoping that the students who did participate will go out and do health or science communication and have a better understanding when they're doing this reporting of what's gone on in the background, to have a better understanding of the language and the methodologies used, and hopefully create better stories from it.
Once we had a better understanding of what we got ourselves into, we were able to come up with a hypothesis. If journalists are embedded in the scientific lab, they will be able to communicate the scientific research more clearly to the general public.
Will the public believe more the scientist or the journalist? I think the journalist. I think it was a very interesting experience.
A very interesting experience indeed. One moment, we would be doing the science while trying to comprehend the reasons behind each step. The next, we would be in journalist mode, documenting the process. It got a bit confusing at times. I'll be the first to admit that.
Okay, quick, Nate, explain what we're doing.
On the spot like that.
Go.
However, once we got into a routine, we were able to capture the meaning behind the science as we were doing it.
Okay, so what we're doing right now is we have a material inside the heat block here, where it's at 42 degrees Celsius, and what that's doing is it's helping the plasmid with the genes inside of it move into the yeast cells.
And like any great scientist, we experienced failure when it came to some of our lab results.
Did our experiment today. We were here for 5.5 hours or so getting ready because we didn't really get the results that we were 100% hoping for. I guess it also helps us realize how tedious this experiment is in all science in general. Not a complete loss, right?
It builds on the Missouri method of learning through doing. And this is something that our journalism students haven't had a chance to do before. Getting into a lab to begin with was kind of groundbreaking when we started it back 4 years ago. Getting a student to do an experiment is a whole other leap from there. So I believe this will help.
We just finished today, so we're done with the lab process of actually doing experiments in the lab. Did that actually help us understand what we were doing? I don't know. It's a little bit shaky. We did a lot of things, but what exactly we were doing was a lot of times that was a little murky. And potentially that might have actually obscured the general context of what we were trying to do. So this is something that we're probably going to have to go home and sit on and let that congeal in our minds and try to pull ourselves back to a wider perspective.
From what I can tell, all the students who participated this time really enjoyed getting to see what the scientific method was about, to see that experiments were a laborious process. It was not you step into the lab and within a week all of a sudden you have discovered something brand new. It's a lot of hit and miss, a lot of repetition, whether it's pipetting or planting or whatever.
The experiment was an eye-opening experience and helped us understand the complexity of science reporting. While the duration of the project lasted about a month, we concluded that more time in the lab would have been beneficial.
So now that we know that there are some things that are achievable, now we can move a little bit up or a little bit longer. So we're going to do it again. We're going to tweak one or two things, but we definitely want to do it again.
In the lab setting, it's about the science, and the traditional thing is, I don't care who communicates it. I don't know if there's an importance in communication aside from getting it into my conferences or my scientific journals. Don't care about the public. Well, the way funding is working now, you have to care about the public. Broader impacts are becoming huge. And to do those broader impacts, you have to be able to explain your research to the public and to those lawmakers that are deciding how much funding does NSF get.
There are multiple ways in which we went about sharing our results. Our initial goal from the beginning was to present our findings at the poster session during Life Sciences Week at the University of Missouri.
And we stamped it on each one, and as you can tell, like when it's just normal, everything grows and everything's fine. But over here, our protein right here didn't grow, which means that the yeast took up the cadmium, and the yeast died.
Aside from presenting a poster, we publicly tweeted and Instagram parts of our experiment, which we later compile in a storified blog post on the SciXchange website. Oh, and of course, by producing this comprehensive video as well.
I'm really excited that it worked because I really didn't know. I wish I could share that excitement with you guys when we saw colonies in those plates. We saw that all the time, but for me it was really exciting to see it because it works.